Good afternoon and welcome to this live session for the course Human Molecular Genetics. I hope all of you are going through the lectures and finding this course to be useful. So we have um, two live sessions for this course, the one that is being done today, and then we'll have one more 2 weeks from now. And apart from that, we have this discussion forum wherein you're free to post your queries and we'll be able to answer all your queries. And of course, a Google spreadsheet was shared with the students, and we have encouraged students to write the queries. And there are some queries that I will address them. And in the live session as well, that you have the chat box where you are free to write your queries and we'll be able to answer. So what I would like to state here is that as an instructor, I would be attending to your queries on the topics that we have covered. Will not take up any questions that relates to the assignments or exams, when is the exam and things like that. These are logistics that you should post it on the course portal or discussion forum, where the NPTEL team will be able to give you a better answer. I don't want to mix these two because I'm not involved in conducting the exam directly. So therefore, I don't want to give you any information that is not correct that would affect your preparations. So we'll be discussing only on the topics. So first, let me take up questions that are there in the Google spreadsheet. Of course, there's again, as I said, that there is a question on exam date, but I think you should post it on the course portal. As I understand, all these NPTEL courses, the exams are conducted on 2 different dates, 2 different exams. You can choose any one of them, which allows you, maximum flexibility that you should consider. So we'll get into the questions that are there. One of the questions that is posted is that, the rate of transcription and translation. So in the sense that how many RNA, mRNA may be made and how many peptides may be made in prokaryotes versus eukaryotes. This is a question which is very difficult to sort of experimentally test, because when you talk about cell, even if you take a human cell, you have some 40,000 genes, but not all of them are going to express. But maybe if you consider in a given cell type, about 30% of the genome or the genes are expressed, and you have about 10,000 plus genes. And for every autosome gene, you have 2 copies that undergo. So how we are going to study is something that is, is a challenging task. But what people have done is that they have used in vitro methods to, understand the rate at which the mRNA is being made in terms of transcription, or you have a nascent mRNA and try to, get a peptide out of it and then calculate the speed at which the ribosomal complex can add amino acids, right? This is what perhaps the question is about. So what we know is that it varies, but it is like, for RNA pol2, it is known from about 1 kb per minute to about 3 kb per minute. That's the difference. But what you need to understand, it's not necessary that an RNA polymerase loaded on to the double helix makes an RNA, and then until it gets completed, then you would not start again. It's like an assembly line. One complex adds and then transcription initiates. It makes a copy. By the time the copying or making of the RNA is going on, you may have another complex initiating the transcription. So if it is a very long gene, like for example, there are genes that spans about 2 megabase, 3 megabase, 4 megabase, you will have multiple polymerases, copying the DNA. So in that way, you have several copies that are made. And it is not only the mRNA, how many copies are that are made that dictates how much protein will be there. It is also the stability of the mRNA. Some mRNAs are stable, that are there for days. Some mRNAs that are very short-lived, only few hours. So the net effect on how much protein is made depends on the stability of the mRNA as well. And that is something that is given, we believe, by the sequence that is present in the 3' UTR, but and it all depends on what kind of function it does, and why should some RNA stay longer and some others should have a short lifespan, because these are all, again, they are selected for certain specific function. For example, you are talking about some signaling mechanism. So there is a ligand that comes and binds and activates certain signaling cascade. As a result, you have few genes are turned on and you are making making the transcription on. Now the signaling cascade should be on as long as it is required. After that, you should be shut down. That is why you have these kind of signaling mechanisms. Therefore, in such cases wherein the gene regulation is controlled by certain external factors like hormones or even temperature and so on, the RNA that are made are short-lived, right? Because then as long as the signal is there, the transcription would happen, and then few hours the RNA may be stable, it is making protein. But once it is, the system changes or restored, for example, normal Z, you should get back and the gene should not be expressed. So it's a different level. And when you talk about translation, again, it is, it varies. Again, these are in vitro studies people have done. It's a, rough estimate is about 20, 25 amino acids per second that the translation rate can be. And again, you have to understand that the translation in a given mRNA, it is not just only one peptide is being made, but there could be a large number of peptides, one after the other. In the same mRNA can be a template for multiple peptides being made. So it's just like a amplification of the signal, right? So that's what it is. Yeah. And there are questions that talks about what is the scope of this field from research perspective. Well, I mean, this is something that you can ask yourself, like how much we know about ourselves, right? Why we look different. Even the different races look different. The Europeans look different, Japanese, Chinese look different, Africans look different, and Indians look different. South Asians look very similar. What is that? The basis for it? This is one aspect. And why certain, for example, diabetes is more prevalent in India, in the Indian population, right? It's not just India, but even the Indian population settled abroad, they have a very high risk of developing diabetes. And likewise, there are populations that have higher prevalence rate for certain complex disorders. And what makes that happen, right? So these are some of those questions that we do not have an answer yet. Even for, if you consider yourself these days, most of the class are held online. But in a classroom, when we sit together as a students community or the instructor, 30, 40, 50 students sit in the same classroom, and one is having cold, and then obviously when he coughs cold or what, sneeze or whatever it is, maybe 4, 5, 6 of the students in the classroom may get the same infection, but not everyone, right? Now, what then makes the difference, and why some are more at higher risk of getting the cold, the common cold, as compared to the others? So every aspect of our physiology is regulated by, in a way, to some extent at least, if not wholly, by your genome, and the variations in the genome brings about variation in the phenotype. How do you respond to, right? Now, this is something that this is still, with a limited understanding, we are saying that, but we haven't understood what variations, makes put you at higher risk of developing a disease, or that prevents otherwise in certain individuals the disease is something that we haven't understood well, and therefore, human genetics is something that is unique, right? Every individual is different, every population is different, and that's the challenge that you have, and it's going to take a long time. So in terms of research, if you ask me, it's amazing potential that you have, right? Because there are a lot more to know than what you know about the human genome and how does it affect even the normal function. Forget about the abnormal genetic disorders and so on, that is anyway given. So there are a lot more to be done there too, right? So that's the answer for that. So you have really a lot of scope for research, even as a researcher or even as a genetic counselor or a medical geneticist who do counseling. As a profession, you have a lot more to do because we haven't understood the genetic basis of all the disorders. So that really helps us, right? There's another question which talks about, if you establish a genetic counseling center in a small city like Kanpur, what kind of approach or strategy do we come up with? By the way, this Kanpur is not a small city. This is the largest city in the state of Uttar Pradesh. And in terms of population, probably it comes among the top 20 cities in the country. And therefore it's not small, When you talk about any disorders that are related to a population, it is the number of individuals who live there. So if more the population, more would be the genetic burden, disease, and so on. So the question is, what kind of approach strategy you come up with? I really do not understand. If you want to start a counseling, then you have to have the required education qualification and background for you to do that, and you have to be associated with some hospitals because they would be referring the patients to you, and that is what required. You should have hospitals wherein people go for their ailment. They are not going to come directly to you if you're a counselor, and then they would refer you to the counselors, and then you look at the family history and you make a pedigree and come up with that. You can counsel. But if you're talking about clinical geneticist, DNA diagnosis, there's a different approach. Earlier, this was not something that is common. Any genotyping or sequencing or even chromosomal preparations were not something that is common earlier. But with the COVID, every small town now you have a pathology clinic that has got a real-time PCR machine, right? Now things may change in times to come, right? Maybe with the sequencing becoming cheaper, you're going to have even smaller cities, much smaller than Kanpur, can have such labs which help in DNA diagnosis, like what you have seen in case of COVID. That is, if you have, I don't know how many of you have done a COVID test. If you look at it, there will be two signatories for the any COVID report. One is, of course, a pathologist. The other one is somebody's called as molecular biologist. It's become mandatory now. The routine pathologists who, as a training in MD pathology, alone cannot certify such report. That has become so. You can see the future how it is changing, right? So, and then there is another question is, what are the current research areas in human genomics? I don't really get it. It depends on which country you are looking at, what you are looking at. These are all country and population specific, right? That is what you should look at it. So you look at from the population specific, in that context you have to come up with. But yes, I mean, the biggest challenge or even the wish list for any human genomics expert is that, can we identify all the health risks that your genome has conferred on you in the future? Say a baby is born, you extract DNA from the baby, sequence it, will you be able to tell what is the risk factors that the baby carries, right? whether he or she is at higher risk for hypertension, higher risk for any of the other ailments, things like that. That is something that, like a horoscope, like you're able to tell, and what are the challenges that we may face as he or she grows older, right? If that is done, then it helps. For example, if you say that you are at higher risk of developing diabetes, you can suggest a better lifestyle right from the beginning. Therefore, the probability of developing diabetes will become lower, right? Likewise, hypertension, then you bring down the cholesterol level. And there are so many lifestyle modifications that can be done. And this is called as personalized medicine. So the personalized medicine is that, and you need to know what, more often when you talk about personalized medicine, is not just treating the disorder, it is preventing the disorder, right? So that for that to happen, you need to know what risk that person carries in terms of his or health in future, right? So in future, what is in store for you, if you can decipher from the genomic sequencing, that is nothing like that. So that is what everyone's goal is. But for us to reach there, we need to understand the variations that are there in every population. It could be different. And not only that, you need to know which variations or which variant is a risk factor for a given disease or condition. That requires a lot more work. So therefore, there's a lot of scope one can do, right? There's one question that talks about, DNA profiling of a person able to predict some disease. That I already answered, sort of. That is something that still people are working on. It's not something that is completely solved. So because the reason is that finding the genetic variant that predispose an individual to develop certain disease is something like, it's a very challenging aspect, because much of these disorders that wherein you have genes play a role are contributed by multiple genes. When I say multiple genes, these are variants in multiple genes. So every gene has got, a large number of alleles. That's what we call as alleles. Now, which allele and combination of which alleles from various genes uh, increases the probability of the disease that you would develop is something that one needs to study, right? That's something that is coming up. We have discussed that in a couple of lectures. If not, you'll get to know that. But these are very, very difficult, study designs. You will understand that these are called as case control studies, something that the study design will be discussed in the lectures. As you go through the lectures, you will understand how do you do that. The other question is, we know that DNA, RNA, protein machinery, but how these decide the overall character of any individual and so on. So these are all, complex processes, and we know one or two examples where such things have happened. For example, we talk about the X chromosome. You have two X chromosomes in a female and only one X in a male. I'm talking about very normal process that happens in our body. And of the two, if you look at any gene, that is, X chromosome is not a small chromosome, unlike Y, which is very small. X is, one of the bigger chromosomes in our, in our cell. So it certainly carries hundreds of genes. And if both X are active in a female cell, then you're going to have double the amount of protein from any of the genes that is present on the X chromosome being made in a female as compared to a male who may have only one copy of the X chromosome. Now there is an imbalance in terms of how much protein that is there. To take care of that, obviously, the mammals evolved a system wherein one of the two X chromosomes are inactivated, or to be more precise, only one chromosome remains active. If an individual has got three X chromosomes, again, only one will remain active. The other two will be inactivated. Now, this inactivation is done by, as you have mentioned, RNA protein machinery. There's a transcript called XIST, right? X chromosome specific transcript, inactive X chromosome specific transcript that is expressed only from the inactive X, and that binds to that X chromosome. It doesn't leave the chromosome where it is being made. And there are proteins that goes and binds to this RNA, and this DNA RNA protein complex makes that um, um, chromosome inactive, right? That's the process that just gives you an idea how does it happen. And same or similar kind of concepts are extended to the regions where we have imprinting. There are genes that are present in the autosome. We may have two copies of the gene, but of the two copies, only one copy is allowed to express. And which copy is expressed also depends on whether the copy has come from your father or mother, right? Paternal or maternal. So there are some genes that express only if it is that allele is derived from, or it's come from paternal lineage or some maternal lineage. These are allele specific expression. And we know that these are called as imprinting clusters. They're not just one gene, but there are multiple genes that follow this pattern. And they are regulated by, again, these kind of non-coding RNAs. And as a complex, and it is what is called as cis control, meaning, wherever that RNA is being made, that particular chromosome, there is some change that happens, either that region of that chromosome, all the genes are expressed or inactivated depending on the mechanism that you are looking at. So therefore, these are some of the examples, but we'll read a little more when you talk about imprinting disorders. So let's look at some of the questions that are coming up in the chat box. I'm not going to answer any questions on the exams. So you can, you can write to in the discussion forum. The NPTEL team will be able to answer that. There's a question called, why do we consider the conservative, non-conservative mutation as no mutation, even though they too induce change in amino acid? Well, strictly speaking, a mutation is nothing but a change in the DNA, But if that change in the DNA does not bring about a change in the phenotype, we generally don't refer that to as a mutation, right? So this is called as variation. These are called as a normal variations, right? These are called as polymorphisms. That is what explained as SNPs, or single nucleotide polymorphism. So if a variation is not resulting in a change in the phenotype, then you don't generally call that as a mutation, right? That's what in definition. So if there is an amino acid which is highly conserved, and there is a change in the codon, therefore you bring in some other amino acid, it affects the protein's function, then that's a mutation. But if that change does not affect to that extent the protein function, this change is tolerated in the population, you're still normal, then you consider this to be a polymorphism. So that is how you come up with different names, that conservative, non-conservative, and so on, depending on the phenotype. Okay, how the phenotype could be at the level of protein or at that level of the individual. So there's some question, how do we obtain different color-coded chromosomes in spectral karyotyping? This is not that really that every chromosome is having a different color. This is called as pseudo color or false color. So what people do is they basically have the probes, right? So you have chromosome specific probes, chromosome 1, chromosome 2, chromosome 3, and so on. These are nothing but the unique sequences representing these DNA that are derived. And then you label them. The same probe you label with red, and then the same DNA, original DNA that you have chosen for labeling with red fluorochrome, you can also label with green fluorochrome, for example. And then what you do is that you have now these two probes, right? One that is labeled with red, one that is labeled with green for each chromosome. And what people do when they develop this kind of spectral karyotyping is that they mix different ratios of these two colored probes for each chromosome. For example, for chromosome 1, the ratio of red versus green is, say, 1:9. For chromosome 2, it is 2:8. Chromosome 3, it is 3:7, and so on. So there is a difference in between the red versus green. After that, you do the hybridization, and then you take the images using color camera. And when you do that, then you do image processing and then plot the ratio of red versus green, right? And you know what ratio that you are given for chromosome 1, 2, 3, 4 are, and so on. And using this information, you will be able to type that the chromosome that gets the ratio of red versus green 1:9 is chromosome 1. Now, if there is a translocation, part of the chromosome 1 gets fused to chromosome 2 or 3, now the same ratio applies there too. Therefore, the software will give you the color which tells that chromosome 1 part of it has gone to chromosome 3 and so on. So that's what spectral karyotyping is in short, but you can read more. Even it is given in Wikipedia if you go and read. I want to know why do activity questions, which are subjective, is calculated for purpose. I don't know. There is. You are talking about subjective question. I thought these are all objective type questions that is given. What is given as assignments are the one that will be used for grading. Let me put it this way. There are 4 assignments for this course. So, best 3 out of the 4 that you attempted, that score would be used as part of the final assessment. That I assume that makes it clear. I'm not answering any more questions on the exams or hall ticket, any of this, right? Yeah. How do you calculate penetrance or incomplete penetrance, particular pedigree? Okay, it's not very difficult. It's simpler. So what you do is that you look at a family, a dominant. Mostly the penetrance issues comes for dominant disorders. So when you have a family, you look at the parents and first generation, second generation, and so on. And you would find that somewhere there is a generation that skips the phenotype, right? You have it, say, for example, the grandparents have, one of them have the phenotype. They have, say, 2 children, of which only one having the symptoms, other is not. But the person who is not showing any symptoms in the second generation, has, that person has a child who develops the symptoms. That is in the third generations, but that in second generation is not there. So when you have this kind of situation, and you know that there is a issue of incomplete penetrance, so what you do is that you plot the entire family and then identify the minimum number of people who may be carrier for the mutation but not showing the phenotype. So easily you can put that, a dominant pattern you'll be able to identify. When you do that, you will know how many are minimum number of carriers and how many are affected. Now that's the sum. And then the number that is carrier but not having the affected is the, if you, for example, the, 10 people, there are 8 that are affected. 2 of them should be showing the symptoms, but they are not showing. But they are expected to be carriers because, the gene must have gone through them. Then you know that it is 20% of this, family who have got the mutation is not showing the phenotype. Therefore, the penetrance of this disease in this, family is 80%. But you don't generally conclude that based on only one family. You generally look at, 5, 6, 7, 8, 9 families, and then you take a average value to talk about what is the penetrance level for the given disease in the population. There's a question that I learned that chimeras have two blood groups. In that case, won't I don't know which chimeras you are talking about. Um, there could be what you call as mosaics that probably you're talking about. Um, um, but so these conditions are congenital issues, but that would have multiple problems. It is not simply the blood group or anything. It could be multiple issues that could be there. So it depends. Therefore, it depends on case to case. One cannot generalize. Yeah. So can we predict that the particular phenotype of parents or grandparents will be transmitted to the next generation before the baby exists? Yes, I mean, you can calculate, for example, my parents, one of them had, say, a disease that is genetic disease, and I am normal, so I may be a carrier for the mutation. Now, what is the probability that my son or daughter would have the defect and the phenotype? Is something that you can calculate. So there are, depending on what you are looking at, if it is a recessive, it is autosomal dominant, autosomal recessive, X-linked, and penetrance problem. If you have this information, you'll be able to calculate the risk of having the disease. It doesn't say that, sometimes it can be 100% if it is a dominant disorder, right? Or if it is, that can be, but not always. So there is a probability that you can calculate. Yeah. There's a question, what could be the probability the rate of mutation in a child born by IVF through parents are normal? I do not, IVF is, in vitro fertilization, right? So normally we assume that the oocyte of the mother is taken, and the sperm of the father is taken, and it is fertilized in vitro outside the body, and you're going to implant the embryo back into the mother's uterus, right? Now, if that happens, this process should not create any new mutation, theoretically. Therefore, whatever mutation they carried, if any, that would manifest that the IVF is not known to induce any new mutation. Otherwise, it would not be practiced. Okay? Good. Right. There's a question about umbilical cord, which has a potential to treat. The umbilical cord, the difference here is that there is a new field of biology called stem cell biology, right? The stem cells are cells that you can basically induce to differentiate into any of the tissue type that you have in your body. For example, I have very severe, I met with an accident where, fire accident, I lost much of my skin. Now, without skin, I cannot survive. Now, what people do is that they do skin grafting. So wherever your skin is left, they take and then patch it out, other part of the body where there is you lost the skin, and allow it to grow, because you can't even get out of the hospital. You are completely immunocompromised. Now, for that grafting to happen, also that you have to have some minimum level of skin left in your body. But if you have a mechanism by which you can induce the stem cells to differentiate into skin, and that can be used to apply on your body, then you can be saved, right? Now, the umbilical cord is identical to you because it has got the same genetic composition, right? It is basically part of the embryo that developed into you what you are today. And the remaining part is umbilical cord. It's not something that derived from mother's tissue. It is attached to the mother's uterus, but it's not mother's tissue. It has genetic identity exactly like you. So therefore, but at the time of birth, these umbilical cord is just, normally it is discarded, right? But what now people advocate is you store these umbilical cord and use that cells that are there to differentiate, induce, differentiate into any other cell type that, for example, here in this case, skin, that is, it has a medical implications should you require any such thing, because technology is developing. Say, for example, if you are now 22 years, so you're talking about genomic sequencing and things like that. Maybe in 20 years from now, maybe there are approaches developed wherein you're able to induce, for example, the kidney or liver or some of your organs from the stem cells. Therefore, if you need that kind of organ replacement due to some medical condition, if you have the stem cells, you can go and make it. Now, you can use umbilical cord only if your parents stored the umbilical cord when you are born, right? It's normally discarded. So that is why they're saying that you store it. Maybe you have ways and means to differentiate them into different organs, and you should be able to use it. So that's what we're doing here. Somebody is asking hard to preserve. I don't think that it is hard to preserve. That is, but still we haven't come up with umbilical cords. Still we haven't not come up with process or approaches to differentiate that into all other types here. Somebody is asking, how do we receive the notification? It is posted. You should be getting an email as well. It is posted in the group. You can check it. You should be receiving emails as well. Yeah. Mm-hmm. Yeah, there is a question. Cancer is caused due to uncontrolled cell growth. What is the mechanism that can stop, inhibit DNA replication, and so on? So, in terms of drugs that can kill the cancerous cell, we certainly have a lot of advancement. But what is difficult is that will you be able to kill every cell that has this property? So that is where the question is. And not only that, there are other factors that all of a sudden induce a cell which is in dormant conditions to become cancer, right? So these are the challenges that we have, right? Somebody's again the umbilical cord thing. I mean, if you want to preserve any cells, if you want to preserve any, which is routinely done in the lab. For example, this lab has got so many cell lines and there are process, but it has to be kept at very low temperature, right? And there are certain medium that is mixed with the cells and stored. Therefore, the property of the cells are not lost. And you cannot have variations in the temperature and so on. That would limit the potential of the cell to differentiate into any of the cell lineage, or even its viability. It may die if you try to culture. So therefore, the environment that through which that you are planning to store, that kind of infrastructure is what the the difficulty level. But it's routinely done in research labs, but not in clinical settings. Yeah. Someone asked a question whether phenotype only all about physical abnormalities or is include physiological condition. Obviously, when you talk about phenotype, it is not appearance. For example, if I have a genetic condition that doesn't allow me to speak, as long as you talk to me, you don't know that I have a disorder, right? Or I can't hear, but I look pretty normal. But the only thing is keep calling my name, I will not respond to you, right? So that is phenotype. It is not appearance. So phenotype is something that is the final, the functional readout of any gene. Let me put it that way. The functional readout could be, even for example, your ability to, feel the temperature. For example, you have thermoreceptors in your hand. If you want to see somebody's having fever, what do? You put your hand on their face and see whether they have temperature, right? You are able to sense that. Now, there are conditions where there is babies are born without any pain receptor, right? Pain receptor is, for example, if you press, you feel the pain. That tells your body that something that is crushing your finger, therefore you should attend to. And that, there's a receptor, the nerve function. Now, there are babies that are born without pain receptor, where simply, even if they put their hand on fire or something fell over their hand, they will not know. Think about yourself. You will not, you have some accident, some cut, wound, you will not feel anything. It is like you are in anesthesia. So that is a condition, right? So therefore, it is encompassing everything. It is not just the phenotype. Yeah. It can be behavioral disorders, could be autism, for example. There are many, many conditions. Anything that is not normal, that is an abnormal phenotype, right? There's a question that can the nonsense mediated decay mechanism be synthesized artificially? I do not understand the question. If you want to do it some in vitro assays, yes. I mean, if you know what are the proteins that are involved in this process, you can do in vitro assays. But what do you mean by synthesized artificially is something that I don't understand. You may want to clarify your questions. Right. Again, the question on IVF is, can de novo mutation IF? See, de novo mutation is a possible. For example, people get skin cancer, right? The skin cancer happens because of UV rays or some other chemical compound that you got exposed to may mutate your cells that are there in your skin, then it becomes cancerous. Likewise, there could be some mutation that happens in the sperm that you have chosen, or the one that gets fertilized, or the oocyte that you have taken. But the probability that the IVF procedure itself is inducing any change at the genome is less likely, because so far there are no reports, at least as far as I know, wherein the de novo mutations rate are much higher in the IVF babies as compared to normally born babies. There's a question on particular disease, whether it's autoimmune disorder. I mean, I don't recall. You can search it. I don't really remember every disease as to whether it has a genetic origin. Yeah. There's a question whether nonsense mediated decay can be brought to be cure for. Well, I don't know. Even if there is a mechanism by which you will be able to suppress the nonsense mediated decay of the mRNA, whether now it will be, the mRNA will be allowed to translate a truncated peptide. That's your question, whether it will be functional. So it depends on where the mutation is, right? If it is very closer to the end of the peptide, and we know you are done all these in vitro studies to show that if even the truncated peptide is there, it is able to do some leaky function. Therefore, you can rescue the phenotype. Yes, this is theoretically is possible, right? People have attempted that kind of thing. But the question is that, that it depends on where is that mutation, right? So it varies from gene to gene and mutation to mutation. So yes, there are attempts like that, right? Somebody, Kartika Goel, this is my first live session in PTL. Welcome. Yeah. There's a question whether we can apply for internship in your lab, but this COVID time, no one is allowed to enter the campus right now. But yeah, I mean, this is possible when the normalcy is back in the campus where people can apply for internship. There are ways by which you can apply. There is one program called Surge. You can apply through that. But then, the number of students that would be selected for such internship will be smaller. That you would appreciate. We cannot really accept all students who show interest. But then lab exposures and all, now in the current scenario, this is not possible. Yeah, there's a question whether this course can allow me to do genetic counseling. I am not sure. That's something that I don't know whether in India there is a course approved program for genetic counseling. It is there in other countries, but whether in India that is there or not, I don't know. There are some universities that have come up with these diploma courses, but if that is required for you, then you need to look at it. But otherwise, you can always practice because from the knowledge that you have gained, if you're a physician, yeah. Yes, there are no internship right now physically in the institute, right? There is a question, any examples of antisense RNA in our body? Yes, I mean, there are, like I gave you example of the X chromosome, how, yeah, for example, non-coding RNA silences the inactive chromosomes, or expression of that RNA results in the silencing of the extra X chromosome that is present. But that expression of that particular RNA is regulated by an antisense RNA. So there are variety of examples of antisense RNA even in the human body. Yes, MD clinical genetics. Yes, that is from LGPGI. But I'm talking about genetic counselors. Like these are in other countries. They need not be physicians. They do other courses. For example, even human genetics, and then they are professional genetic counselors. They are not necessarily from the medical stream. Yeah, so that's what I was referring to. Yeah. I guess we have pretty much come to the end of this session, and I hope I was able to answer most of your queries. And if you have any more queries, then you can post them in the course portal discussion forum. We'll be able to answer. Um, there's a question: do we need to have coding skills to pursue? See, these are all tools. If you want to learn any programming, these are all tools, right? So that helps you to do something using a different perspective. So that's what it is. So you can always do that. If you have a code, you want to analyze some large data from human genetics, you can always do that. Otherwise, you have to do manually. So every research tools that you learn, if you can learn more, it helps you to do better analysis. So yeah. Loss of function mutation, you explained about founder's effect. What about bottleneck effect? What do you mean by bottleneck effect? What's the value of the human molecular genetic course getting jobs? See, it is not like something like I've done a programming, or I've done I know typing kind. These are specialized branches of science that you learn if you're interested in to either do a research or as an academic, like a faculty, or to become a genetic counselor. So that's the branch that you are looking at. Yeah. Mitochondrial DNA is affected due to particular type of cancer. Will baby also get affected same type of cancer? No, it depends on whether that defective mitochondrial DNA is packed in the oocyte and whether that is transmitted to the next generation, right? Because it is a maternal, the mitochondrial transmission. So it depends on that. Yeah. See, I mean, I don't know. Founder effectively refer to something that somebody carries a given mutation and seed a new population. Therefore, that particular variant is more common. Now, that is why I asked you, what is that bottleneck effect that you are referring to? You are talking about a bottleneck effect is something that reduces the population. And if this reduction is random, meaning, it is not that preferentially removing people of particular phenotype, then you should not see any difference in the frequency of that, allele in the population, theoretically, right? So that should not have. But if the reduction is based on certain, for example, sickle cell anemia, in people where the population is at higher risk of malaria, then you'll find large number of heterozygotes because it confers a protection. But if the bottleneck effect is nothing to do with the allele that they carry, it is a random effect, then you should not alter the frequency of any, allele in that population. Does transposition seen in humans? Yes, of course. There are disorders that are caused by the transposable element, moving and jumping, landing in a gene and making it, defective. And it's a all rare thing, but all these things are, sort of reflected in our genome. And there are many gene duplication that you see in our genome, possibly a result of this kind of transposable elements, because in a gene is there on either side, you have, identical transposable element. Does it leave a copy there? Now that leads to what is called as non-allelic recombination or unequal recombination, leading to one allele having two copies of the gene, one not having any gene copy. Now, when you have two gene copies in the same chromosome, Now, one is doing the original function, the other one now does something new function because that is free from the original function. And it may lose its function or it may give a novel function that is beneficial. People believe that the hemoglobin that you have, fetal hemoglobin, is something that, that evolved in this kind of a process where it acquired, very higher affinity for the oxygen. Therefore, it can capture the oxygen from the mother's blood, right? That people think is because of this kind of a processes, right? No, we don't know for sure, but that's the theory or hypothesis. Okay, good. Then I think we have done for 1 hour. If there are more questions, we'll take it up in the course portal. And thank you very much for all of you to have joined here. And
thank you so much, sir, for your valuable session.
Right.
Can I end the session, sir?
Yeah, please. Yeah.
Okay, sir. Thank you, sir.